Today we are going to discuss on a very important topic in genetics, which is called genome mapping. Genome maps are nothing but they show us the attributes of genome. For instance, they mark as genes. Is similar to geographical maps where we have all the attributes, for example, road, river, mountains. Genome map are similar to this. In a way, the genes and markers are the map, the attributes. Now, there are two types of maps. First is the genetic map. Genetic map is based on the use of genetic techniques to construct genetic maps showing positions of gene and other sequence features on a genome. Genetic techniques include crossbreeding, experiment, pedigree analysis in human being, etc., etc. And physical mapping uses molecular biology techniques to examine DNA molecule directly in order to construct maps showing the positions of sequence featuring sequence features including genes. Now let's go quickly into what are genetic maps. Now and tools that are used to study genetic map. Genes were the first markers to be used in genetic mapping. For a gene to be a marker, it is useful, it is important that there must exist in at least two forms or two alleles. So one of the main drawbacks is that when we deal with certain genes which had more number of alleles, it becomes very cumbersome. So gene was less favored. But the first genetic map in the early 20th century, in the early decades of 20th century, genetic mapping was done using genes as markers. Then came the era of DNA markers. So all the map features that are not gene are DNA markers. It has some advantages over genes as markers. So using DNA marker, genetic mapping can also be done. There are three important tools that are used to study genetic mapping: restriction fragment length polymorphism, RFLP in short, simple sequence length polymorphism, SSLPs, and single nucleotide polymorphism. We'll quickly go through one by one. As we can see, RFLP is restrictionization of fragments of DNA to study polymorphic restriction sites. So as we can see in this particular slide, there are two alleles of one gene, say for example, and in allele number 1, there are 3 cuts. Allele number 2, there are 2 cuts. So the number of fragments in allele number 1 is going to be 4, and number of fragments in allele number 2 are going to be 3 fragments. Now all of us know that restriction enzymes recognize a particular site in the genome on which it acts and cuts the DNA fragment. So this is how RFLP based on the length of the polymorphic DNA mapping can be done. Now RFLP can be assessed in 2 different ways. RFLP can be scored in 2 different ways. Number 1, sudden hybridization of the DNA fragments. Number 2, by PCR analysis. So once the DNA is restricted using restriction enzymes, the DNA is run on agarose gel. It will show us as a smear. Now those smears can be transferred to nylon membrane, and this membrane can be used to using a probe to find out the number of fragments in the experiment. Similarly, PCR also can be used to assess or to score the RFLP, where primers are designed on the polymorphic sites, restriction sites. So, and a PCR is done. Depending upon the number of cuts, it will show us different number of bands in the PCR product in agarose gel electrophoresis. So, can quickly show you the picture here in the screen. So, sudden hybridization and PCR. If there is a cut, if there is no cut in the fragment, then it will show us as a single band. If there is one cut, it will show us as a double band, 2 bands. Similarly, in PCR, since the PCR primers are designed in the restriction site, polymorphic site, depending upon the number of restriction sites, it will show us as a form of band in our agarose gel electrophoresis. This is about RFLP. Now let's move to simple sequence length polymorphism, SSLP. SSLP are areas of repeat sequences that display length variation. So different alleles containing different number of repeats as units. There are 2 types of SSLPs, mini satellite and microsatellites, depending upon the number of units, repeat units. Microsatellite is more preferred over mini satellite for various number of reasons, for which I will not go into the details at this stage. I'll quickly show you how the typing is done in the case of SSLPs. We all know SSLP is nothing but the tandem repeats of DNA. So let us consider 2 alleles, allele 1 and allele 2, and you can see it here. There are 3 repeats of ZA. Nucleotide ZA repeat 3 times in allele number 1, and in allele number 2, ZA repeats are repeated 5 times. 5 times. So a PCR primer can be designed along with this repeat sequence and the flanking regions to amplify the region based on the number of repeats and amplification products. All the amplification products can be run on the agarose gel electrophoresis. Depending upon the size of the number of repeats, the bands can be seen in the agarose gel electrophoresis. The picture on the right shows us the agarose gel electrophoresis as an example of lane A and lane B, where difference in the size is shown here. So likewise, with the number of repeats, more number of repeats, the DNA band becomes bigger. So size can be ascertained accordingly. So this is how SSLPs are basically done. Then single nucleotide polymorphism. These are positions in the genome where some individuals have one nucleotide and others have a different nucleotide. Say in one Z, in other individual it's C. So this is what single nucleotide polymorphism stands for. As we can see it in allele number 1 and 2, there is a difference of the position Z and C. At one position, allele 1 has G, allele 2 has C. So this is what single nucleotide polymorphism stands for. This have been used to study genetic mapping. Now, one of the most important factor in genetic mapping is that the frequency of recombination. So recombination frequency is very important. This is the basis for genetic mapping. Now, in other words, the recombination frequency, the frequency with which the genes are unlinked by crossing over will be directly proportional to how far they are on the chromosome. So this is the basis for genetic mapping. Now, recombination frequency of different pairs of genes, using recombinant frequency of different pairs of genes, we can construct a map of and find out the relative position in the chromosome. This is the basis for genetic mapping. However, genetic mapping has some shortcomings. Number one, the resolution of genetic map depends on the number of crossing over that have been scored, and sometimes genetic map have been found to be limited accuracy. Now, one of the most important finding was that after the sequencing of chromosome 3 of Saccharomyces cerevisiae, it was found that physical map and genetic map were different in two very important genes. In two very important genes, which was found to be in genetic map, it was found to be incorrect compared to physical map, further determined by DNA sequencing. That is the reason why physical mapping is now preferred. So I will quickly go through some of the important techniques for physical mapping now. Number one, restriction mapping, which locates the relative position on a DNA molecule of the recognition sequence for recognition endonucleases. Fluorescence in situ hybridization, in which marker location are mapped by hybridizing a probe containing the marker to intact chromosomes. And then sequence tag site mapping, STS mapping, in which the positions of short sequences are mapped by examining genomic fragment by PCR and or hybridization. So these are three important techniques for physical mapping, and in today's class we will be looking into restriction mapping in details. So as I was telling you, genetic mapping using RFLPs as DNA markers, they can locate the positions of polymorphic restriction sites within a genome. But very few of the restriction sites in a genome are polymorphic. So many sites are not mapped by the technique. They are not represented by this technique. Then people started thinking, could we increase the marker density on the genome map by using alternative method to locate the position of some of the non-polymorphic restriction sites? That was the basis for restriction mapping. So this is what restriction mapping achieves. Now, so we go into restriction mapping now. The simplest way to construct a restriction map is to compare the fragment sizes produced when DNA molecule is digested with two different restriction enzymes which recognize different target sequences. For example, in today's class, we will be looking into an example where both EcoR1 and BAMH1 enzymes are utilized to perform a restriction mapping. So what happens is, first the DNA is digested using one of the enzymes, and the size of the resulting fragments are ascertained using agar cell electrophoresis. Next, the DNA molecule is then digested with the second enzyme, and the fragments are again sized in agar cell electrophoresis. So far, the result we have gotten by the two individual digestion, we are able to enumerate the restriction sites and numbers of each of the enzymes. But we do not still are in the position to find the relative positions of the restriction site. Now comes the most important digestion in restriction mapping, or what is called double digestion, whereby both the enzymes are simultaneously used to digest the DNA, the original DNA. Now, when this type of experiment is carried out, and when we are dealing with a longer fragment, sometimes it becomes so important, and sometimes it becomes so important that there are two possible alternatives, two alternate maps come into picture. We'll see to that in the next slide. So in that way, only double digestion is not appropriate. It's not complete. It's not useful. We also perform another set of digestion which is called partial digestion or incomplete digestion, whereby the digestion of the DNA with one of the enzymes is performed in suboptimal conditions. Suboptimal conditions, so that the product is not fully digested. So we'll see the importance of that. Now let us take an example of a 4.9 kilobases fragment for which we are going to perform a restriction mapping. Let us consider two enzymes, EcoR1 and BAMH1, to be used in this particular experiment. Now, when 4.9 kb fragment is digested using EcoR1, there are two fragments are produced. 1.5 kb fragment, 3.4 kb fragment. In another set of experiment, when BAMH1 is used to digest 4.9 kb DNA, there are four fragments were produced: 0.7 kb, 1 kb, 1.2 kb and 2 kb fragments. Now when both EcoR1 and BAMH1 were used for double digestion of the same original DNA, 5 fragments were produced: 0.2 kb, 0.5 kb, 1.2 kb, 2 kb and 1 kb. Now we are going to find out the restriction site of the two enzymes. In other words, we're going to map the restriction site of these two enzymes. So let's go into that. Now before we go into how we do maps, how mapping is done, let us quickly go into the situation where digestion was done in a suboptimal condition, or in other words, incomplete digestion or partial digestion was performed. When BAMH1 was used to partial digest the 4.9 kb fragment, there were many fragments were produced: 1.0, 1.7, 3.7, and so many other fragments, which you can see it in the screen. Now, using this, we are going to map the sites. So this particular slide shows us how mapping is done. First, 4.9 kb fragment is digested using EcoR1, BAMH1 separately, and then both EcoR1 and BAMH1 together, double digestion. Now let us try to interpret what does the fragment say. Fragment 0.2 kb and 0.5 kb in the double digest. In the double digest, there were two fragments, 0.2 and 0.5. So what does it mean? Looking at the other two fragments, the other two products of EcoR1 and BAMH1, this 0.2 and 0.5 kb fragment must derive from 0.7 kb BAMH1 fragment. 0.7 kb BAMH1 fragment with an internal restriction site of EcoR1. In other words, the BAMH1 fragment, which was 0.7 kb, also has an internal EcoR1 site, and EcoR1 has acted upon it, and it has become 0.2 and 0.5 separately. This is the possible interpretation of 0.2 and 0.5 kb. Now, if we draw a map, we can clearly see it in the slide. If we draw a map, 0.5 kb and 0.2 kb, altogether 0.7 kb with one internal enzyme cut, EcoR1 cut site. So B to E is 0.5 kb length. Now E to V is 0.2 kb. So altogether 0.7 kb. This is done. Second, 1 kb fragment, which is a product of double digestion of EcoR1 and BAMH1. Now this BAMH1, this must have this 1.0 kb fragment must have derived from BAMH1 fragment. There was 1 kb fragment in a BAMH1 digestion. So this 1 kb in a double digestion is also from from BAMH1 with no internal EcoR1 cut site. Now we can account for the 1.5 kb EcoR1 fragment. We can look at the EcoR1 digestion where two fragments were produced, 1.5 kb and 3.4 kb. Excuse me. So now we can construct a map of this 1.5 kb of EcoR1. So 1.5, we can see it on in the slide also. We already have accounted for 0.5 and 0.2, 0.7 BAMH1 site. Now the 1.5 kb EcoR1 site, EcoR1 fragment could be 1 kb 1 kb fragment from the EcoR1 plus BAMH1 product, digestion product. So we have as we can see it here, 1.5 kb can be constructed in the way it has been assembled here. Now, so far the story is clear and very simple. So we have mapped this EcoR1 site, we have mapped the BAMH1 site till now. Now there are another two fragments, 1.2 kb and 2.0 kb. Now this must also be the BAMH1 fragment with no internal sites, and they must lie within the 3.4 kb EcoR1 fragments. Now there are two possibilities now. As we can see in map 1 and map 2, the 0.7 fragment is already there, we can see it. Now 1.2 and 2, either 1.2 can come in between, or 2.0 can come in between. In other words, after 0.7, 1.2 and then 2 kb. Similarly in map 2, 0.7, 2 kb, 1.2 kb. So we need to find out which one is the correct orientation, which one is the correct map. For this kind of situation, the digestion products of suboptimal condition, for example, or partial digestion, comes into the rescue. Now, you can see here, if map 1 was correct, then the partial restriction product will include a fragment which is 1.2 kb plus 0.7. Since it was BAMH1 restriction digestion, so BAMH1 site is at 0.7 and 1 at 1.2. If map 1 is correct, then we should expect in the partial digestion, we should expect a fragment of size 1.9 kb. Similarly, if map 2 was correct, then the partial digestion product should include a fragment of 2 kb and 0.7, that is equivalent to 2.7 kb. So partial digestion is basically performed to find out some of the uncuts. So in this particular picture, we can see from the partial digestion that a 2.7 kb was already present, but not 1.9, which means map 2, which has a position of 0.7, 2 and 1.2 is the correct map. So we can map the relative positions of restriction sites of the enzyme. So this is how physical mapping is done, and this is an example for restriction mapping. So this is all for today, and thank you all. Thank you very much.